Hello and welcome to Module 6.1. In this module we're going to talk about cell signaling, and this information is covered in textbook chapter 9. So we're talking about cell signaling, which can also be called cell communication. And in multicellular organisms, what we see happening is that cells receive a very large variety of signals from other cells. When they get these signals, they can have several responses, including changing their metabolism, their shape. They can be told to grow and divide, or even to die via apoptosis. They can also be told to migrate, which is moving, adhere to a surface, or actually differentiate into a different cell type. So in this lecture we're going to be talking about the different types of signals, the different types of receptors, and the signaling pathways that are most common in the cells. This figure gives an overview of a cell surface receptor, and the most common type of signaling pathway is shown here. We start with reception. This is where a primary messenger, which we'll also call a ligand is just a general term for a molecule that binds to another molecule. In the first step, called reception, we have this primary messenger which binds to a receptor on the target cell. Then the next step is called signal transduction. So after the primary messenger has bound to our receptor here, the receptor does what we call signal transduction. Signal transduction is the receptor converting the primary messenger into a secondary messenger. Finally, the third step is a response. So the responses can be many, like we just talked about, but they can be lumped into two main types of responses. The first is an activation of cellular responses. This is like a change in metabolism. And the second is a change in gene expression. So this is where DNA is transcribed into an RNA and translated into protein, and we can either upregulate or downregulate the transcription of DNA. Upregulating would mean we would make more transcripts, and downregulating would mean we are making fewer transcripts. There are four main types or forms of cell signaling, and these are based on how far away the cell producing the signal is from the cell receiving the signal. The first and closest form of cell signaling is called autocrine signaling. This is where the producing cell, or the cell producing the cell signal, is also the cell receiving the signal. So it's basically signaling itself. This can also extend to signaling very similar cells close by. The second form of cell signaling is called signaling across what are called gap junctions. This is where two cells are connected to each other. And a gap junction is a transmembrane protein that has a water-filled channel that allows signals to move from the cytosol of the signaling cell directly into the cytosol of the target cell. Next, and getting even further away, is called paracrine signaling. This is where molecules from the signaling cell are exported and they simply diffuse locally to the target cell. One example that we've talked about is neurotransmitters, where a presynaptic neuron will release neurotransmitters into the synaptic cleft, which is a very small space between cells. These neurotransmitters will then diffuse across the synapse and bind to the postsynaptic cell, which in this case would be the target cell. So this is paracrine signaling, which is local signaling via diffusion of signals. The final form of cell signaling, and the one that works the furthest distance away, is called endocrine signaling. This is where a signaling cell releases a ligand into the bloodstream, which then moves throughout the body to the target cell. Endocrine cells originate in glands such as the thyroid gland, the hypothalamus, and the pituitary gland. And the ligands that they release are called hormones. There are two main types of receptors: internal receptors and cell surface receptors. Cell surface receptors are going to be what we focus on in this lecture. However, Internal receptors are also important. So an internal receptor does not require the transduction of a signal across the cell membrane by a transmembrane protein, because the signaling molecules are hydrophobic, and they can simply diffuse across the plasma membrane. Remember that the more hydrophobic and the smaller a molecule is, the easier a time it has diffusing through the cell membrane. So these guys just simply diffuse into the cell where they bind to an intracellular receptor. An intracellular receptor is a receptor that is in the cytoplasm of the cell. These signals are often transcription factors that will interact with DNA in the nucleus to regulate gene expression. And the way they regulate gene expression is the signal molecule will bind the intracellular receptor. That signal molecule intracellular receptor combination will then diffuse into the nucleus and promote the transcription of DNA into RNA. The second type of receptor is called a cell surface receptor. This is the receptor we talked about in our general form of cell signaling, and this is what we are going to focus on in the rest of this lecture. Cell surface receptors are transmembrane proteins, which means that they have an intracellular domain, a domain that exists inside the cell membrane, and an extracellular domain. And the way these receptors work is that a ligand, which again is our primary messenger, will bind to the extracellular domain of the protein. The protein will then change conformation and transduce that signal into the cell by activating secondary messengers. And what's important to note here is that the ligand never actually enters the cell. It simply binds the extracellular portion of our cell surface receptor and causes a change in that receptor. This way it can dissociate from the cell surface receptor and move on to the next cell and activate it. So one ligand can activate multiple cells and multiple receptors because it's not used up by being taken into the cell. Another important feature of cell surface receptors is that they are specific to the ligand. So oftentimes a single cell surface receptor will bind to a single ligand. And because of this, we have many different types of cell surface receptors expressed inside our cell membrane. There are three main types of cell surface receptors. The first is an ion channel linked receptor. This is an ion channel that binds a ligand which changes the conformation of the ion channel from closed to open, allowing for ions to flow down their electrochemical gradient into the cell. This is the type of signaling we have already talked about in, again, neurotransmitters, wherein the signaling molecule is a neurotransmitter, which crosses a synapse, binds to an ion channel linked receptor, which then goes from closed to open, allowing ions in, which starts another action potential in the postsynaptic cell. These types of receptors are made up, again, of an extracellular and intracellular, and also a domain that is inside the cell membrane. Many of the amino acids in the membrane spanning region are hydrophobic in nature because they are exposed to a hydrophobic environment, whereas the amino acids that are in the extracellular and intracellular domains are often hydrophilic amino acids because these are exposed to water, whether it be in the cytosol or in the extracellular domain. The second main type of cell surface receptor is called a G-protein linked receptor. These are heterotrimeric G proteins, which means they have three different subunits. We call these subunits alpha, beta, and gamma. The way these works is that a signaling molecule will bind to the G protein, as seen here, causing the GDP molecule that is associated with the three subunits to dissociate or unbind, and a GTP will bind. The binding of GTP causes the gamma and beta subunits to dissociate from the alpha subunit. Dissociate just means that their binding is changed and they actually are no longer bound, they are separated from each other. This separation causes the cellular response. The cellular response is the activation of secondary signaling molecules, which again change metabolic processes or change the way DNA is expressed. Next, the GTP will be hydrolyzed. So this hydrolysis reaction is one that uses water, and this reaction will remove a phosphate group from the GTP, turning it into GDP, which then gets us back to where we started with alpha, beta, gamma subunits bound to GDP and the Gprotein coupled receptor. Third type of receptor, cell surface receptor, is called an enzyme-linked receptor. We're going to look at one called a receptor tyrosine kinase. So this is two subunits, or two individual units of the receptor, that when signaling molecules bind, these two units come together and dimerize, which then allows them to phosphorylate the tyrosine residues on the inside of the cell, the intracellular domain. And after that phosphorylation event, the protein can then cause a cellular response here. So this enzyme-linked receptor is different than the other types because the enzyme, or the receptor itself, is an enzyme or is closely associated with an enzyme. Whereas with the other types of receptors, the receptor and the enzyme are separate entities. So we've talked about the types of receptors. Let's talk about the type of signaling molecules. The first type are small hydrophobic molecules. Now, because these are small and hydrophobic, they can easily pass through the cell membrane. So these don't need a cell surface protein receptor. They can actually just diffuse straight into the cell where they can interact with our internal receptors. So these will go into the cell and cause the reaction, whether it be a change in metabolism or a change in how DNA is expressed without the use of a cell surface protein receptor. Here are a couple of examples. These are based on cholesterol, or sterol, and these are defined by this structure here. There are 3 6-carbon rings and 1 5-carbon ring. That's kind of the basis of a steroid. You can see here testosterone and estradiol. These are 2 common hormones that we see in our body. The second type is water-soluble ligands. So these are water-soluble, which means they're polar, and they're often large, so they are not going to pass through the cell membrane without some help. So these guys usually bind to our cell surface receptors on the extracellular domain and then have the signal transduced to the inside of the cell. This is a very wide and diverse group of ligands. These include small molecules, peptides, and proteins. Finally, there are gases that act as signaling molecules. Nitric oxide is a gas, not to be confused with nitrous oxide, which is what makes your car fast and furious. But this is actually a molecule that will diffuse through the membrane and cause smooth muscle cells to relax. This is very important in blood vessels, causing them to dilate or expand and restoring blood flow to the heart or other organs. So I'd like to go one step further, or one layer further in this process, and look at the propagation of a signal. And we're going to take, for our example, EGF. EGF is a enzyme-linked receptor. It's a receptor tyrosine kinase. This is involved in the regulation of cell growth, wound healing, and tissue repair. So what happens is when EGF binds to the EGF receptor, we have what's called a signaling cascade, or a signaling pathway of downstream events that cause the cell to grow or divide. So this is a very powerful signal. If the epidermal growth factor receptor is activated at inappropriate times, so if it's activated when it shouldn't be, this leads to uncontrolled cell growth, which we call cancer. And the important concept here is that after the EGF receptor is activated, it starts a chain reaction of events that eventually leads to a change in the cell's environment. And let's get into a little more detail on what that chain reaction is. So what happens here is when EGF binds to the receptor, we have two proteins that are associated with the receptor, GRB2 and SOS. So GRB2 and SOS then activate a molecule called RAS, which then activates RAF, which then causes MEK to be phosphorylated. And after MEK is phosphorylated, it will phosphorylate ERK. And now we have this active phosphorylated form of ERK that does two different things. It enters the nucleus and triggers a cell response. So it triggers the translation, or the transcription and translation of specific genes that cause cell proliferation, migration, growth of new blood vessels, and they inhibit programmed cell death. These signaling pathways can get pretty complex, but they're all based on one molecule activating another, which then activates another, so on and so forth, in what we call a signaling cascade or a pathway. So when we have a molecule like ERK that goes from an inactivated form to an activated form, this can be done several different ways. And one of the most common ways to activate a molecule in these signaling pathways is by phosphorylation. So that's the first method of intracellular signaling we're going to talk about. Phosphorylation is simply the addition of a phosphate group to a molecule, like a protein. And here we have some examples. This is a serine molecule where the hydroxyl group has been replaced with a phosphate group. It's common to replace the hydroxyl group. And if we're going to transfer a phosphate group through the action of an enzyme, we call that enzyme a kinase. So whenever you see the word kinase in an enzyme's name, you know that that enzyme catalyzes the addition of a phosphate group. We often have a lot of different types of second messengers. Second messengers are small molecules that propagate a signal after it's been initiated by the binding of a signaling factor. So these are intracellular proteins. And one of the most common is called cyclic AMP. You can see that this starts with ATP, loses two phosphate groups, and then you have one phosphate bound at two spots to this pentose sugar, and you'll get cyclic AMP. So this is a common second messenger. And the main role of this messenger is to bind and activate an enzyme called cyclic AMP-dependent kinase. So here we know that if cyclic AMP is activating this molecule, which is a kinase, that means that is going to phosphorylate other second messengers or other proteins. So in this way, a kinase regulates a lot of different metabolic pathways. So we've talked a lot about molecules like ERK becoming activated, going into the nucleus and changing the way that DNA is transcribed into RNA, but we haven't really gotten into how that's done. And so this is one of the main signal endpoints or responses to the signal, and that's gene expression. I'd like to go into a little more detail on how this works. So in our previous example, we had phosphorylated ERK, and we said it goes into the nucleus, causes some sort of change to DNA transcription. Well, how it works is, a lot of times you have part of your mRNA, so this is the 5' end of our mRNA. This is called a 5' UTR, or untranslated region. And you can see here that the mRNA has actually formed sort of a loop where it is bound to itself. You can imagine that this would block any sort of ribosomal activity from moving down the mRNA, because it's going to hit this 5' UTR structure and be unable to move past it. So ERK is a kinase, so what it's going to do is phosphorylate this molecule called MNK1. Now the specific names of these molecules aren't important at this point. Just realize that there are many different proteins involved. And then through a cascade of phosphorylations, we're going to have this group of proteins work together to unfold that 5' untranslated region, which then allows protein synthesis to occur. Another big response to cell signaling is the increase in cellular metabolism. For this example, let's take adrenaline. Adrenaline is also known as epinephrine, and it's the fight or flight hormone. So this is produced in the adrenal gland, which is attached to your kidney. So it's going to release epinephrine into your bloodstream. Releasing a molecule into the bloodstream means this is endocrine signaling. So it's going to activate receptors on the cell surface, especially in muscle cells. It's going to lead to an increase in our second messenger called cyclic AMP. The cyclic AMP is going to start a signaling cascade, and the end result is the activation of two enzymes. So we're not talking about a change in the way genes are expressed here. We're talking about the way that enzymes are activated inside the cytosol. The first enzyme is called glycogen phosphorylase. This is going to catabolize glycogen into glucose. So remember, in the muscle cells, we have these long strands of glycogen, which are just repeated glucose molecules. This glycogen phosphate is going to break those up, so we have individual glucose molecules. And of course, we can then take those glucose molecules into glycolysis and get energy out of them. You're going to need some energy when you have an adrenaline rush, because your body thinks to itself it's time to either fight or run away, and that means your muscles are going to have to do some work, so they're going to need energy. The second thing it does is it turns on a protein called glycogen synthase, and this actually inhibits the formation of glycogen from glucose. So you can imagine, if you start breaking glycogen down into glucose, you don't want to then immediately start making glycogen again. You want that glucose to remain free. So glycogen synthase is going to prevent the formation of glycogen and allow that glucose to be free inside the cell, so that your muscles can use that glucose to produce energy. So the third response a cell can have is cell growth. This is started or activated by growth factors, which are very, very strong signals. So cells don't normally divide unless they are told to do so. Cells in their resting state are not prone to divide. So if they get a growth factor, that's actually going to tell the cell that it's time to grow and divide into two cells. One of the last things that a cell can be told to do is to die. This programmed cell death is called apoptosis. And this allows the cell to die in a controlled manner that prevents the cell from breaking open and releasing the molecules that are inside the cell, which can potentially cause an inflammatory response. Well, that's all for this lecture. It's important to know the different types of cell signaling, the different types of cell surface receptors, the different kinds of cell signaling molecules, and it's important to understand the different responses that a cell can have to a signal. So I appreciate your time, and thank you for watching.